The next speaker is Tarun Kumar. He is from the HP Labs. So actually, I had invited my colleague, Dr. Manikandan Narayanan from IIT Madras, Computer Science Department. Unfortunately, he was not in town today. So Tarun recently finished his PhD jointly with Professor B. Ravindran and and Professor Manikandan. So he will be actually presenting work which he did with Dr. Manikandan at IIT Madras. Also, I'm happy to inform that tomorrow is the IITM convocation and Tarun gets the Best PhD Thesis Award in Data Science. So Tarun, thanks for accepting and looking forward to your talk.
Thanks to all the organizers and thanks for the generous introduction. So I'm Tarun Kumar. Currently I am affiliated with HP Labs, but the work that I will present today, that was mostly done when I was at IIT Madras as part of the Robert Bosch Center for Data Science and AI. And this work is done with the Intel fellowship that I had during my PhD with Dr. Ramanathan Sethuraman. So today's agenda of this talk is I'll begin with the introduction to complex graph structures. So by training, I am from computer science background. So all the methods that I will present, they'll have all the bias that I obtained during my training. And then I'll talk about some of the applications which you saw in the title. So I'll begin with an introduction to all the complex graph structures that are out there, and then we'll zoom into one structure which I'll present in detail, and which is a multi-layer network. Then I'll show how multi-tissue systems can be modeled as multi-layer networks, and then I'll show its example in finding node centrality in multi-layer networks with two case studies to discover hormone-gene relations and analyze some topological shift in AD disease population. And in the end, I'll talk about all the future possibilities that can arise from this work. So I know there is a format for this presentation. At the end, we have questions and discussions, but please feel free to stop me at any time. I have very few slides, so otherwise I'll finish in like 10 minutes. So please feel free to stop me in between. So let me see if the pointer also works. Okay, great. Just to begin the introduction, if there are people from computer science background in the audience, even if there aren't, so we have seen all these structures like at the top left corner, knowledge graphs. So Google is famous for their large knowledge graphs. Even when you search things, you get some prompt from Google where they, at least publicly they say that they create a knowledge graph and from there they create it. So this is in reality a complex graph, right? So there are nodes which have types, there are edges which have relations, and overall they represent some information, and it is easier to query this representation as compared to some unstructured format like documents and web pages and so on. I will not talk about this graph today. And then there is another complex graph structure called multiplex graph, where multiple layers represent the same system, but the aspect that they capture is different. So here you can see this is actually a transport graph on some area in UK, where the nodes, they represent cities, and there are edges between nodes if there is a particular mode of transport possible between these cities. So for example, they can be rail, coach, air, and so on. And then there is a multi-layer graph, which is more general version of multiplex graph, where you have multiple modalities of interaction, but there is also a cross interaction possible between layers. So you can see that node 2 in layer 1 can interact with node 3 in layer 3. So, and the last one I want to talk about is hypergraphs, which capture the superdyadic relations between entities. So at least I think many of you would have seen how protein complexes exist, right? So they are not just dyadic interactions. So multiple proteins make these complexes. And today's talk, I will focus on multi-layer graph. But I spoke about all these four so that if you have any questions, ideas later, you can reach out to me and we can have discussions around them. So in multi-layer graph, first we'll see that why do we want to study this. So our bigger agenda is that we want to understand multi-tissue system. To be particular, we want to understand how multi-tissue how communication happens in multi-tissue systems. So for example, if you see here, there are 3 tissues in this system, and tissue ABC, and all tissues have same set of genes, but they have some different interacting patterns. So these can be gene regulatory networks, or maybe gene-gene co-expression networks, and so on. So in this system, we want to understand which genes contribute to inter-tissue communication. Okay, anyone wants to guess in tissue C what genes are more likely to derive inter-tissue communication? Any guess? When is tea break after this? Okay. G2 and G1, because G2 has 2 connections that go out of that tissue, and G1 has 1 connection that goes out of that tissue. That's what you want to say, right? You are not saying. Okay. Okay. After G1 and G2. Okay. If I say between G3 and G5. Okay. Between G3 and G5, which is a better candidate? G3. Why? It has no connections outside. Okay, then you already know the remaining work that I'll present. No, but that is the point here. So the genes which upfront, they may not look like they have interactions outside that tissue. Maybe the data, or even when we model the data as this network, we have all the methods have their own limitations. All the experiments that are done, they have their own limitations, and they are sometimes not. Even the communication does not always happen with the interlayer connection. So they can spread within one tissue, and then they can go beyond. So yeah, you are right. G3 is a better candidate than G5, because from G3 the impact, if I can use the term, it can go to G2, and from G2 it can travel out. But from G5 there are lesser chances. So we want to identify such genes in these multi-tissue systems, which may not have direct connections outside that tissue, but still they are good candidates to derive inter-tissue communication. And we use centrality measures to do that. Now, how do we first come to this network, this multi-layer network? So we begin with this multi-tissue gene expression data. In these experiments, we used GTX data, where we have data which is collected from lots of individuals. So this is a 3-dimensional tensor. Each layer represents a tissue, and the columns represent genes. And when we represent this data as a network, genes become nodes, all the tissues become layers. So if this was the actual data, three layered, we'll get these 3 tissues. So every layer maps to a tissue, and from this, sorry, yeah. Yep.
How do you account for that?
Yeah. So in this work, this is a very valid question. In this work, we did not account for that, but having said that, there are some factors which we took care of. So even in this gene expression data, there are lots of confounders present. Even there is a variability because of different different categories in the population and so on. So we took care of that, but this is mostly averaged over all the cell types. Here we don't go to the level where we have single cell data and so on, but that is a really interesting direction. We thought of it, but there is lack of data, so we could not reach that level. And another reason is because one of our goal was to come up with method more, so data becomes like something which is given. From there, how do we make a model which can find all these hidden relations? Okay, so this data can be represented by 3 tissues, 3 layers, and because we want to capture the impact of genes which goes outside tissues. So in a way, we are also saying that we want to capture the impact of genes which stays within tissues, right? So we want to characterize this within and across tissue effect. So for that, we represent this multi-tissue network by super-adjacency matrix, we call it. Similar to adjacency matrix, but it has a special pattern. And we split this into two super-adjacency matrices. where one captures only within layer connections and one captures all the across layer connections. So I'm just using these terms M, A, C here because I'll use them extensively. But just remember that A is something which has within tissue connections, C is something which has across tissue connections. And in real world, if I can take an example, so think of this system where we have two tissues, pancreas and muscle. And we know that in pancreas, insulin is produced, and muscle, there are genes in muscle that respond to it. So let's say there is a set in pancreas that is known to produce insulin, and there is a set of genes in muscle which responds to it. And if we know one set which produces insulin or secretes insulin, we want to know which set of genes is highly likely to respond to it, and vice versa. So and that answers our question: which genes are more likely to participate in intertissue communication. So to do this, when we started working on this, we thought, okay, there may be one centrality equation that can just find this impact of a gene which goes outside that tissue. But when we started working on this, we came up with lots of equations. And what they capture here is, so each gene has a local impact which is limited to only that particular tissue. That is captured by this top equation. It is very similar to page rank equation, if people are aware of that literature. And if instead of A, which only captures within layer connections, we also use C, it represents something called global centrality. So if you don't want to look here, just by looking at this figure, every gene, let's say gene 6 here, it has some impact which goes only to pancreas tissue, which is given by this top equation. It has some impact which goes to every other tissue as well, not just pancreas. If there are other tissues, then it goes there as well, which is given by global centrality. And if we want to condition its impact on particular layers only, which are not pancreas, then there is target layer centrality. And this is the most interesting centrality, which is query set centrality, which captures the impact of a node on another node which is in another layer. So for example, how likely is that this gene number 2 will respond to insulin which was produced by this set at the top. And so when we did our experiments, we begin with the multi-tissue data. From there we get the multilayer network. We split the matrices. There we derive all the centrality equations. And in the previous slide, I showed the centrality equations as they are in bullets, right? As if they are separate equations, but they have relations between each other. So they make a tree of equations. And at any layer in this tree, like if it is a layer specific centrality for different layers, if we sum up all the layers, it goes back to the global centrality and so on. So we have some theoretical results. I'll not talk about those here, but we do prove all the convergence of these iterative equations and so on. Now to validate these equations, one form of validation is where we use ground truth. So you remember, we know what genes secrete insulin. We want to know what genes react to those insulins, and vice versa. So if we know one side of the equation, we want to know whether we are able to discover the other side of the equation. So these are the results that we get for hormone production or hormone responding genes. So if we are predicting insulin producing genes, it means we know what genes respond to insulin and so on. So this is a recall at K curve, higher the better. I am showing only 4 hormones here. Reason 1, because our results are better on these 4 hormones. Our second reason is because one thing which impact our experiments is how much ground truth data we have. So when we know insulin producing genes, if we know only very few genes like 2 or 3, there is no chance to predict genes that respond to insulin. So we want large number of genes on either side to predict one or the other. So here you can see that y-axis that goes to 160, but in some of the hormones it goes to 16 and 14. As this number goes low, the performance of our model goes down. So that is one thing that is still open, that if someone of you wants to take it. I'll be happy to discuss. So this is the form of validation which can be done when we know the ground truth. But again, our ground truth is limited, and for many hormones we don't know the ground truth. And there we use different forms of analysis on these results. So for example, the genes that are ranked higher in insulin, higher in pancreas, which respond to insulin, we did a disease enrichment analysis to see that if there is if they are enriched for any disease which is related to insulin, and we saw some of the good results, like for insulin pancreas there is diabetes mellitus, which is expected to come at the top. So another form of analysis that we have done is, again, when there is no ground truth, how can we use literature to see whether the gene hormone associations that we are predicting, they are present in the literature or not. So we came up with two forms of validation here. One is just to look at those gene hormone pairs and see whether their co-occurrence is better than random. So this number has to be greater than 1, this number has to be greater than 0, then it is a good finding. So in most of the cases we have seen that our numbers our results make sense. But I think the work that Professor Nirav Bhatt he presented, this form of analysis, we did it 2 years back when ChatGPT LLMs were not viral. But now I think they can be done in really fast way when you can just feed all your PubMed data and you can ask, you can generate a prompt which can just give you what gene hormone relations are present there. So that can be another possible directions which people can take. And I want to briefly talk about another application where we tested this multi-sense framework. All the results that I showed you till now, we only spoke about 2 layers at once, but there can be any number of layers. For example, in this brain network, there are 4 regions, and the setup is same. We have same set of genes and different connections between them. And here our idea is, if we know what are the some query set of genes, in this case synaptic signaling genes, is there any change in the topology of other genes present in this multilayer network between control population and AD population? And our results show that there is a noticeable shift in their centrality scores, and this is a more relevant slide where we do the path enrichment analysis, and we find that in diseased population there are some they are enriched for some particular category of pathways. Control population, they are enriched for some other pathways. So this is pretty much that I had. And yeah, in the end I want to conclude this, that we worked with these multi-layer networks. We presented, we came up with multi-sense framework which can find node centrality in multi-layer networks. And it has numerous applications. We tested it on 2. But one interesting thing is that when we started working on this multi-sense framework, we were actually focusing on transport networks, where you have different transport modalities as different layers, and you want to know the impact of one modality on the other junctions in another modality. But again, there was lack of data, so we could not proceed with that, and here we found really good applications. But that is again one avenue where this work can be extended. And yeah, all the other complex graph structures that I spoke in the beginning, how centrality frameworks like this can be extended to those is also an open question. Yeah, so thank you. All the code is available on GitHub, you can use. We are working on coming up with a web portal where you can just give your query set and get done with all the analysis. Right now, the code is in Python, so if you want to use it, you can contact me on these email IDs. You need some knowledge of Python, but in short time, we'll also come up with the web portal. So yeah, thank you. So.
Thanks, Tarun. Questions? We have a lot of time.
Hi, Tarun. Great talk. Really enjoyed it. I was curious, like this brain network that you showed. Yeah. So I can understand that this within layer links essentially correspond to maybe gene interactions of sometimes. But what do the across layer links indicate?
Yeah, so all these multi-layer networks, even this one and the other, the gene hormone relations when we predict. So we start with gene expression data, right? So when we say within layer, then they are gene-gene interactions, and we do some correlation calculation, and from there we get it. Even across layer, the same framework follows. In this figure, I think it will be more clear. Here. So let's say the first layer here, the one at the front is tissue 1, and the second is tissue 2, tissue 3. So the first column here, and maybe another column in another layer, which represent another gene in another tissue. How well they are correlated, that is what we capture with this within across layer connections. One thing that we have observed here is our networks are highly sparse when we talk about across layer connections, but there is lot of interaction that happens because of this. So this is across layer communication, right? There has to be some link. If not for that particular gene, maybe for other gene, there has to be some link to go there. And there are some of the very recent works where people have started taking this across layer connections into account. So at least, actually before we started working on this, there was a work where people only looked at direct connections between one layer and another layer genes and so on. But they represent the same gene-gene interaction. In this case, they represent how well co-expressed these two genes are, maybe in different tissues. But this is very sparse, highly sparse.
Right. So in the brain context, sorry, Arijit, can I have a follow-up? Yeah. So in the brain context, I mean, I understand that what you're looking at is correlation, not necessarily causality.
True.
But given that different parts of the genes are, like for example, processing different sensory modalities, there could be completely different reasons why certain genes are switching on. So is it possible to do something like your ground truth testing for the brain data set to just see that those links, that across layer links which you have obtained through correlation are actually indicative of some causal relation?
Okay, I'll answer this in two steps. One, we did some, we took care of some confounding factors while making these networks, but you are correct, these relations might be of some effect which is not really what we are looking for here. So for these gene hormones, we are working with a group where they are going to test some of our associations. On the brain networks, we have not worked with anyone yet, but this is a good point, I think, at least to check whether the links that we are observing, they should be there or not, right? In short, yeah.
This one. Sorry, I can't resist asking this, but it's a completely different thing, not biological at all. But long back, I mean, one of the first works on this multi-layer networks is the famous work by John Paget, who actually looked at the relations between the various influential families in Renaissance Florence. And what he was looking at was 3 layers. One layer was how the families were related through marriage relations. Another was how they were related through business dealings. The third was how they are supporting each other to get political office. And what he showed was that the Medici, which became the most powerful family in Renaissance period, they were not central in any of the individual layers. But once he looks across the layers, they essentially turned out to be central. So I'm just curious, have you thought of actually applying this new centrality measures that you have discovered on that old data set?
Okay, we have not applied it there, but I have seen some studies along the similar lines. So all the multi-layer network analysis was initially done in social sciences groups, and people have worked on identifying or locating terrorists in some regions to which character is important in Star Wars, like all these applications. So yeah, there are lots of works there, but we have not applied this particular centrality framework there. Having said that, I think one important application of centrality in multi-layer networks, which I think can answer lots of questions in these social sciences, is betweenness or closeness centrality, where if you take only one modality of interaction, the person in social sciences, the person might not be very central, might not be close to things, or might not be between things. But when you talk about multi-layer networks, things change, dynamics change. That is what. Yeah, thank you for that.
Yeah, so in continuation of the previous discussion, so if you look at tissue 1, 2, 3, and you are looking at this correlation of genes across, do you see that mostly the housekeeping genes are the ones that are really showing up, and then that really is not a true representation of a cross-tissue interaction?
Yeah. They do. And one way we get rid of it is when we, so the genes which are housekeeping genes, they are likely to have more connections. Because they have more connections, our centrality framework will be biased to rank those, not necessarily biased, but it will rank those genes higher. So when we analyze our results, at least the plots that I presented, we get rid of all those genes. So that is a non-housekeeping genes ranking. They come at the top, but they are towards the top.
So I was just curious, what is the physical proximity of these tissues that you are looking at?
There are different hormones that we have tested here. There are some tissues which are closer to each other, but there are many, especially when we talk about tissues like hypothalamus and some tissue near stomach or maybe, right? So they are not close to each other, but for some hormones this is the combination that works, right? So there we have seen that so results make sense there as well. Yeah. So physical proximity will give you denser networks sometimes. But if they are far away, your network may become sparse.
So maybe somewhat similar question. So have you tried to look at intercellular communication in the hormones? Because I think a lot of hormonal regulation is happening that way. So have you tried that?
Yeah, true. Continuing to discussion with Professor Sinha. The data that we started with is averaged over multiple cell types. But if so if you look at this multi-layer structure here, where we have 3 layers, every layer can be thought of representing multiple cellular layers, right? So if there are, let's say, 10 cellular types, then this will result in 30 layered structure, where 10 layers represent one tissue, and so on. So whether the framework that I have presented will extend to that or not is I need to check, but we have not looked at it, at least in these experiments.
Talking about adding another additional layer, for example, apart from tissues, if you're talking about adding like a social layer or even modality like different omics, how do you envision that happening here? Would it come under another a layer on top or dimension on top, or would it be a function of the genes?
So one thing which we have done here, I did not talk about it, is something very similar. So all the gene expression data that we have here, that results in this these gene-gene interactions, which we encode by calculating their correlation. But we superimpose this network with another network, which is protein-protein interactions, which we get from Stanford Laboratory SNAP dataset, it is called. And we just impose that data here. And what we have observed is that that data, SNAP data, it has very few number of genes across all the tissues, very few number of connections. But those connections have very high confidence. Right. So that data alone cannot answer the questions that we want to ask in the end. This data alone is susceptible to noise and other things, and it has its own challenges. Across layers are sparse, within layers somewhere it is dense because of housekeeping genes sometimes. So we combined these two data sets just by superimposing them to create another network, and we worked with that. But if I talk about having something like where one layer represents connections or interactions within a particular cell, there I don't think that, that's again I'm just thinking out loud. I don't think that can be another layer here. We have to come up with another hierarchy of layers where multiple cellular layers make one tissue, and these tissues are again multi-tissue system and so on. Yeah.
So, is this data, so actually there are 3 different layers, right?
Yes.
So if these, the data for these different layers come from different experiments, so how do you account for the heterogeneity of that data?
Yeah, so we use GTX portal to get this data, and GTX portal actually shares these additional vectors where they encode that even the samples that are used for sometimes different different experiments are different. So we remove that impact. We correct our data to take care of that. And even there are sometimes some hidden factors as well. So GTX actually shares some, I believe they are principal components of the data, and it shares I think many, I think 30 or 40. We set our data for top 5, because one problem is if we correct our data for many confounding factors, we lose signal as well. So there is a trade-off between these two. So we did experiment with that.
We are seeing correlation of expression of one gene with another gene in another tissue.
Yeah.
And we say that one might be hormone and that might be causal for the expression of the gene in other tissue.
Sorry, I could not follow the second sentence. So we are looking at expression of one gene in one layer, one tissue, expression of another gene in another tissue, and we look at their correlation.
Correlation.
Yeah.
So
Next sentence was?
here in tissue 3 and tissue 2, 2 is expressed in like 2, the node 2 is expressed in all 3 layers.
Yeah.
So do you account for like the same gene being expressed, or do you exclude the genes that are expressed in both tissues and then do the correlation? I mean,
Okay.
was my question clear?
Let me answer it, then just let me know if I am answering the correct question. Okay. So you are asking if there is one gene which is present across tissues, and if there is a gene which is not present across tissues, how do you work with this scenario? Then I was going to answer another question.
Okay.
Yeah. Please
So
ask again.
then 2 is expressed in all tissues.
All tissues, yeah.
But we are looking at correlations between expressions across tissues.
Yes.
So do you account for 2 being expressed in all, because
Okay. So actually, we look at only those genes which are expressed, not top expressed, but we have the expression data for those genes in all the layers. So one condition in this multi-layer network is the number of nodes is the set of nodes is same across layers. If there is to answer this, if there is a gene here, which maybe because of our stringent cutoffs or the way its experiments were done, if it happens that it has no connections anywhere, not within layer, not across layer, we keep it just to make our mathematical framework work there. So we keep it. Still, I'm not sure if I have answered the right question, but partial marks.
Any further questions? We have time for one more question. Okay, so then I have a question.
Yeah.
How does the measures that you have proposed for multi-layer networks, how would they be extended in your view to hypergraphs?
Measure, okay, so the centrality, the centrality framework that
I would actually revisit that question. I will first ask the following question. So there are various centrality measures for graphs.
Right.
Which of them, according to your knowledge, have been extended to hypergraphs?
Yeah. So there are many PageRank based centrality or similar to PageRank. Even the one that we have extended is similar to PageRank. The works that I am aware of, there are some betweenness centrality based works where they are extended to multi-layer networks, even to hypergraphs. And there are these works on Shapley value based centrality. They are extended to hypergraphs, I know, not sure if they are extended to multi-layer networks or not. But to extend anything which is, extend any framework from graph to either multi-layer graph or to hypergraph, the semantics that we have to take care are the following. For the work, for the frameworks which are related to PageRank centrality, so they are essentially some random walk measures, right? So there is a random walker and you are biasing you are introducing a bias to visit some nodes more frequently with some different probabilities. In case of hypergraphs, you have to take care of this at the superdyadic level. Like how do you account for maybe larger hyperedges? How do you account for smaller hyperedges and so on? So you have to answer this question there. Then you are solving it for hypergraph. Here you have to answer the question: how can it jump from one layer to another layer? What derives that jump? So in all the centrality equations that I listed down, only this jump varies. When sometimes you want to quantify the impact of one gene on another layer, maybe on another layer, but only some genes in another layer. So that jump probability varies. So this is how the random walk based centrality measures will extend. But there are other centrality measures like betweenness and closeness and so on. There the path matters. And how do you take care of one hyperedge to another hyperedge relation? And in this case, how do you treat within layer connection and across layer connection in terms of path? That will decide how you can extend them to different modalities, different structures.
My last question is, so if I think of, let's say, metabolic networks, it is very clear that bipartite formalism or hypergraph is a better picture framework to model it.
Yeah.
So for tissue-tissue communication, according to you, what is the best framework, multilayer or hypergraphs?
It depends what do you want to call a node. So in this case, if a hormone is also a node, right, and genes are also nodes, it is a bipartite, right? Mm-hmm. Not completely though, because genes can interact with each other, right? So in bipartite that cannot happen. So we have actually worked on combining multilayer graphs and hypergraphs in not with an application in biology, but in some product graphs from Amazon dataset. But we could not succeed much, so that was just a workshop paper. We could take it to that point. But one problem which we faced there was there is not enough rich data set to construct something which can be multilayer as well as hypergraph at the same time. So and the reviewers also said that. So this. Yeah.
Thanks, Tarun. So I think we need to thank Tarun for patiently answering so many questions. Right?
Thanks.
Thank you, Tarun.